Nitrogen is a critical limiting element for plant growth and production. It is a major component of chlorophyll, the most important pigment needed for photosynthesis, as well as amino acids, the key building blocks of proteins. It is also found in other important biomolecules such as ATP and nucleic acids. Atmospheric nitrogen cannot be used directly by plants and animals. Atmospheric nitrogen has to be converted into usable form of nitrogen, and this is done by a process called biological nitrogen fixation. This process is found only in some prokaryotes and is catalyzed by the nitrogenase enzyme complex. These organisms utilize the enzyme nitrogenase to catalyze the conversion of atmospheric nitrogen N2 to ammonia, that is NH3. Plants can readily assimilate ammonia to produce the nitrogenous biomolecules. Bacteria containing nitrogenase occupy an indispensable ecological niche, supplying fixed nitrogen to the global nitrogen cycle. Due to this inceptive role in the nitrogen cycle, diazotropes, that is, the nitrogen fixing organisms, are present in virtually all ecosystems. They are seen in environments as varied as aerobic soils such as azotobacter species, the ocean surface layers, for example trichodesmium, and specialized nodules in legume roots, that is Rhizobium. The reduction of atmospheric nitrogen is a complex process that requires a large input of energy to proceed. In any ecosystem, diazotropes must respond to varied environmental conditions to regulate the tremendously taxing nitrogen fixation process. All characterized diazotropes regulate nitrogenase enzyme at the transcriptional level. A smaller set also possesses a fast-acting post-translational regulation system. Although there is a little apparent variation in the sequences and structures of nitrogenase's enzyme, there appear to be almost as many nitrogenase regulating schemes as there are nitrogen fixing species. Regulation is described on a molecular basis, focusing on the functional and structural characteristics of the proteins responsible for control of nitrogen fixation. Microorganisms that fix nitrogen require 16 moles of ATP to reduce each mole of nitrogen. These organisms obtain this energy by oxidizing organic molecules. Non-photosynthetic free-living microorganisms must obtain these molecules from other organisms, while photosynthetic microorganisms such as cyanobacteria use sugars produced by photosynthesis. Associative and symbiotic nitrogen fixing microorganisms obtain these compounds from the rhizospheres of their host plants. Nitrogen fixation takes a great deal of energy. If the conditions are not favorable for nitrogen fixation, or if there is enough ammonia around, nitrogen fixing bacteria turn off the production of proteins needed for nitrogen fixation. The NIF genes are responsible for the coding of related proteins and associated with the fixation of atmospheric nitrogen into a form of nitrogen available to plants. These genes are found in nitrogen fixing bacteria and cyanobacteria. The NIF genes are found in both free-living nitrogen fixing bacteria and in symbiotic bacteria in various plants. Nitrogen fixing protein production is regulated by the NIF Regulon. The NIF Regulon contains factors which both turn on and off the production of proteins needed for nitrogen fixation. Now, what is meant by a Regulon? It is a group of genes that is regulated by the same regulatory molecule. The genes of a regulon share a common regulatory element, binding site or promoter. The genes comprising a regulon may be located non-contiguously in the genome. The NIF regulon comprises 7 operons, which include 17 NIF genes. Operon is a unit of genetic material that functions in a coordinated manner by means of an operator, a promoter, and structural genes that are transcribed together. The NIF genes are genes encoding enzymes involved in the fixation of atmospheric nitrogen. The primary enzyme encoded by the NIF genes is the nitrogenase complex, which is responsible for converting atmospheric nitrogen to other nitrogen forms such as ammonia, which plants can use for various purposes. Besides the nitrogenase enzyme, the NIF genes also encode a number of regulatory proteins involved in nitrogen fixation. The expression of the NIF genes is induced as a response to low concentrations of fixed nitrogen and oxygen concentrations. The low oxygen concentrations are actively maintained in the root environment. Nitrogen fixation is regulated by NIF regulon, which is a set of 7 operons which include 17 NIF genes. NIF genes have both positive and negative regulators. Some of the NIF genes are NIF A, D, L, K, F, H, S, U, Y, W, and Z. Activation of NIF genes transcription is done by the nitrogen sensitive NIF A protein. When there isn't enough fixed nitrogen factor available for the plant's use, NTRC, which is a RNA polymerase, triggers NIF A's expression. NIF A then activates the rest of the transcription for the NIF genes. If there is a sufficient amount of reduced nitrogen or if oxygen is present, another protein NIF L is activated. In turn, NIF L inhibits NIF A activity, which results in the inhibition of nitrogenase formation. NIF L is then regulated by other proteins that are sensors for the levels of oxygen and ammonia in the surrounding environment. NIF L is also regulated by the products of GLN-D and GLN-K. The NIF genes can be found on bacteria's chromosomes, but many times they are found on bacteria's plasmids with other genes related to nitrogen fixation, such as the genes needed for the bacteria to communicate with the plant host, such as Nod genes. Regulation by oxygen. The action site of oxygen is the NIF L protein, which is basically a flavoprotein with FAD as the redox sensing cofactor. FNR, that is fumarate nitrate reduction regulator, is the signal transduction molecule which transduces the oxygen status to the NIF L protein. In the absence of oxygen, the NIF L protein is in its reduced form with FADH2 as the cofactor and is unable to inhibit the action of NIF A protein. In the presence of oxygen, oxidized NIF L with FAD as the cofactor inhibits NIF A protein and thereby turns off all the other operons. Regulation by ammonium ions. The presence of ammonium ions in large amounts in the environment inhibits the transcription of nitrogenase and all the other NIF genes. Ammonium ions acts as a co-repressor to the glutamine synthetase by modifying it covalently by adenylation. This modified enzyme binds to the NIF-R region of the NIF-RLA operon and prevents the transcription of the genes NIF-L and NIF-A. So, there is no initiation of transcription of the other genes by the delta RNA polymerase. Regulation by GLNK protein. In the nitrogen limiting growth conditions, the inhibition of NIF-A protein by the NIF-L protein is prevented by the antagonizing action of GLNK protein on the NIF-L proteins. A homologue of the NIF-L and NIF-A regulatory gene system has not been found among the eukaryotes. The expression and regulation of NIF genes, while sharing common features in all or most of the nitrogen fixing organisms in nature, have distinct characters and qualities that differ from one diazotrope to another. Here I would like to give a few examples of NIF gene structure and regulation in different diazotropes. Klebsiella pneumoniae. It is a free-living anaerobic nitrogen fixing bacterium. It contains a total of 20 NIF genes located on the chromosome in a 24 kb region. NIF-H, NIF-D and NIF-K encode the nitrogenase subunits, while NIF-E, NIF-N, NIF-U, NIF-S, NIF-V, NIF-W, NIF-X, NIF-B and NIF-Q encode proteins involved in the assembly and incorporation of iron and molybdenum atoms into the nitrogenase subunits. NIF-F and NIF-J encode proteins related to electron transfer taking place in the reduction process, and NIF-A and NIF-L are regulatory proteins in charge of regulating the expression of the other NIF genes. Rhodospirillum ruberum. It is a free-living anaerobic photosynthetic bacterium which, in addition to the transcriptional controls described above, regulates expression of the NIF genes also in a metabolic way through a reversible ADP ribosylation of a specific arginine residue in the nitrogenase complex. The ribosylation takes place when reduced nitrogen is present, and it causes a barrier in the electron transfer flow and thereby inactivates nitrogenase activity. The enzymes catalyzing the ribosylation are called DraG and DraT. Rhodobacter capsulatus. It is a free-living anaerobic phototroph containing a transcriptional NIF gene regulatory system. R. capsulatus regulates NIF gene expression through NIF-A in the same manner described before, but it uses a different NIF-A activator which initiates the NTRC. NTRC activates a different expression of NIF-A and the other NIF genes. Rhizobium species. It is a gram-negative symbiotic nitrogen fixing bacteria that usually forms a symbiotic relationship with legume species. In some rhizobia, the NIF genes are located on plasmids called symplasmids. Sym means symbiosis. Symplasmids contain genes related to nitrogen fixation and metabolism, while the chromosomes contain most of the housekeeping genes of the bacteria. Regulation of the NIF genes is at the transcriptional level and is dependent on the colonization of the plant host. Nod factors act as signaling molecules between symbiotic bacteria and plants in response to flavonoids secreted by legume root hairs. Rhizobium refers to bacteria that include Rhizobia, Bradyrhizobia, Azorhizobia, and Cynorhizobia. Members of these genera are capable of forming endosymbiotic interactions with leguminous plants. These bacteria have the ability to fix nitrogen into a form that plants can utilize. The enzyme that is used for this process is nitrogenase, and it has the ability to reduce atmospheric nitrogen to ammonia at normal temperatures and atmospheric pressure. Nod factors are secreted by Rhizobia during a tightly regulated signaling pathway, which results in the formation of root nodules in leguminous plants, resulting in the Rhizobia becoming bacteroids. Nodulation process. Nodule development. It takes place in several steps. First, the leguminous plant releases flavonoids into the area surrounding the plant roots known as the rhizosphere when the available nitrogen in the soil is depleted. Then the rhizobia in the surrounding soil secrete Nod D, which is a protein that recognizes the flavonoids secreted by the plants. Now, interaction with the flavonoids activates Nod D, and Nod D returns to the rhizobium to induce the transcription of Nod ABC genes. Nod ABC proteins modify the Nod factor in response to the flavonoids released by the plants. The Nod factors interact with the plant and cause root hair curling and root hair deformation. This leads to the formation of an infection thread into the root hair. Nod factors cause cortical cell division in the primordium of the plant and form a nodule. The rhizobia move through the plant root hair via the infection thread to the nodule within the plant. Rhizobia switch into bacteroid form and are surrounded by a plant-derived symbiosome membrane. Rhizobia begin nitrogen fixation and provide the legume with amino acids such as glutamine and asparagine, or ureates such as allantoin and allantoic acid, which are made in the nodule. The legume provides simple sugars produced from plant catabolism to the rhizobia in the nodule. Nod factors are the important signaling molecules in the symbiotic interaction between rhizobia and leguminous plants. When they bind to the root hairs of the leguminous plants, they cause root hair deformations, activation of plant genes, initiation of cortical cell division, and nodule formation. Each of these processes are necessary for the proper formation of root nodules, which enable the symbiotic interaction between rhizobia and leguminous plants. Nod factors control the specificity of the interaction between the two organisms, and they specifically cause morphological and physiological changes in leguminous plants. Additionally, Nod factors can affect other plants in the soil surrounding them. When Nod factors are present in mixed crop fields, they have the ability to stimulate seed germination, promote plant growth, increase photosynthetic rates, and increase grain yields of legume and non-legume crops. Thus, Nod factors do not only benefit leguminous plants, but can contribute to the growth rate of many different types of crops. There are two modes of specificity for Rhizobium legume symbiosis. The first is the interaction between the flavonoids, the chemical signals that are derived from the legumes. The recognition of the flavonoids by the bacteria is dependent on the interactions with node D. When node D is activated, it leads to the expression of node ABC as well as many other node genes, which results in the structural modification of node factors. The second mode of specificity is dependent on the structure of the node factors themselves. Since there are some bacteria that can make several structurally different node factors and therefore have different plant hosts, slight variation in the node factor structure leads to host specificity. Thus, we have discussed how biological nitrogen fixation is regulated by NIF genes and nodule formation regulated by node genes. In the next module, we will discuss the process of nitrate reduction and ammonia assimilation.